Hello Year 10 and welcome to your lesson while I am away on this Friday. What I'd like you to do, as same as always, we are going to have a starter task. You can see on the board we have a diagram of the national grid. What I want you to do is describe the role of each part of this national grid. So think about what happens at the power station, the transformers, the pylons and wires, and also at the consumer. This is a 5-mark question, so you'll have roughly 5 minutes to do so. There is also an extension question which is talking about why is it an efficient system, okay? Think about what those bits in red letters are on that diagram. That's going to give you a clue on how to answer that. The cover teacher will now press pause and give you 5 minutes to complete this task. Okay, the cover teacher would have pressed play again by now. I'm about to show the marks, okay, or the mark scheme so you can green pen your work. Remember to put in the margin of your page how many marks out of 5 you get. Okay, here is the mark scheme on here. So you can see that the power station, this is where electricity is produced. Then you've got our step-up transformer, which is where you increase the voltage, which then decreases the current. Okay, this means that less energy is wasted as heat through the cables. The pylons and cables were where we transport that electricity at that high voltage. And then the step down is the opposite for the step up. Okay, so this decreases the voltage and then therefore the current. And this means that it's safer for use in homes. And then you've got the consumer, which for example is a home. You can also have that for factories, but the voltage is lowered to 230 volts for homes in the UK. Again, this is just to make sure it's safe and there is no risks of electrocution. Make sure you green pen your work. If you need to pause this, ask the cover teacher and then you can spend a little bit more time making sure you've got this all marked. Remember, put how many marks you get in the margin of your page. So today's lesson, okay, is going to be about food tests. There would be usually some required practicals for this part of this lesson, but unfortunately we cannot do that at the moment. You will see we've got our learning intentions for the lesson here. The first of which is we're going to state the positive results of the 4 tests, okay, that we're going to learn about. We're going to also describe how to test for the presence of each different food type for these 4 tests. And then we're going to compare these methods, okay, for all of them. So thinking about, we're going to learn what these 4 tests are, what the positive tests will be, and then we're going to compare those methods slightly. So the food types that we're going to be testing for, okay, include carbohydrates, proteins, lipids, and also sugars. Okay, there are slightly different methods for these. There is a link on this slide, which the cover teacher will also have in the cover sheet document. Okay, they can play this for you if you still do not understand after my explanations using this PowerPoint. Okay, so if you feel like you need to watch the video as well, let the cover teacher know, and then they'll be able to put that on as well as my description in this video. So the aim for this sort of practical, okay, if we were to be doing this in front of each other, we would be testing various food groups, okay, for the presence of carbohydrates, including starches and sugars, okay, proteins and lipids, okay, lipids also known as fats, okay. You will see we have 4 different types of chemical in front of us, okay, and these solutions are what we're going to use to test for those different types of food. So the start of our method, what we would do is we would grab our food sample, okay. You can see on the left side of the slide here, this is called a pestle and mortar. What we would do is put our food sample in the bottom, okay, grind it up into very, very small pieces, okay. We would transfer that as almost a paste, okay, to a beaker, okay, and then add some distilled water, and then that would then make that food sample dissolve into that water, okay. We would then filter that solution, so you can see that in this diagram here, we've got some filtered paper, okay, and this is a funnel. So we would then filter that solution to remove any suspended particles. So any bits of sort of that food left over would be caught, okay, in the filter paper, and then we would be able to use those. So you can see from here, we will put our sort of food samples, 2 centimeters cubed of each, okay, and then we're going to put them in 4 different test tubes. This will then be what we're going to test for each of the different food types. Okay, so you can see for test 1, we're going to have starch. The next test, we're going to have sugar. The next one, we're going to have protein. And then last of all, we're going to test for lipids. Okay, remember lipids are the same, so also another name for fats. So which test, well, what sort of solution are we going to use to test for starch? That's going to be iodine. For sugar, we're going to use Benedict's solution. For proteins, we're going to use what's called a Burette solution. And then for lipids, we're going to use Sudan 3 solution. Okay, so here you can see each different test, okay, and the solution we are going to use for each. We will then put a certain amount of those different solutions into the test tubes, okay, and then if they are present, a certain change would occur that then we could see and mark that down in our practical. So what would we do for each of these tests? So for test tube 1, we would put our starch in. You can see that's over here. Okay, we're going to just add a few drops of solution and we're going to record our observations. If it goes a dark sort of brown color, that means that starch is present. Again, next one along, we're going to think about testing for our sugars. Okay, that's this one here, so Benedict's solution. We're going to add a roughly 10 drops of that, and then you also have to leave it in that test tube and then pop it in a water bath for 5 minutes. Okay, that has to be a certain high enough temperature. And once again, we will record our observations. Protein, okay, we're going to use Burette's solution. We're going to only add 2 centimeters cubed of this, which we'll be able to measure using our pipettes. Okay, once you've done that, you will gently shake and once again record your observations. And then for this last one, for fats, okay, or for lipids, we're going to use Sudan 3, add 3 drops of that. And again, very same as the protein one, we're going to gently shake, okay, and we're going to record our observations. So what I would like you to do is now, based on these previous slides that we have been learning about, I want you to answer these true versus false tasks. So the first sentence is focusing on, is iodine used to test for starch? If we think that's true or false, pop that down. Number 2, Benedict's solution is used to test for lipids. Number 3, Burette's solution is used to test for proteins. Number 4, at the start of the practical, you crush up the food samples. And then number 5, when testing for starch, you leave it in the test tube of water, or in water, sorry, for 5 minutes after adding the iodine. So if you think these are true, just put a T statement right next to them. If you think they are false, try and correct them using the information we've had on our previous slides. I'll give you a couple of minutes to do this. The cover teacher will now pause this and then play again when you're ready to move on. So this next part of the lesson, what I would like you to do is write a short 2 to 3 step method for each test. Okay, there's going to be images on the next few slides that have a rough sort of graphic version of what that test would be like. Make sure that you include the positive tests, okay, for each different food type. So thinking about these, we need to make sure that we know what the color change or that change would be, so we can see if that certain type of food type is present for our food test. Okay, so maybe just a couple of bullet points for each one. Make sure you write the name of each food test, okay, and then make sure you include what that positive test would look like. Our first one, we are going to be testing for carbohydrates. Okay, so starch is a carbohydrate. You can see in this diagram, we have our food sample in our test tube. We are going to add iodine, okay, via a pipette. If the color remains a browny orange, the starch isn't present. So you can see here, that is the color of the iodine itself. And then if starch is present, it goes to a blue-black color. Okay, you have to remember that phrase blue-black because that is what they're going to be looking for in your exams. So make sure you get your 2-step method for this. So I would say add iodine to a food sample. Then your next bullet point would be if color remains browny orange, starch isn't present. And then your third bullet point, if color changes to blue-black, starch is present. If you need to pause this or replay, raise your hand and let the cover teacher know. I'll give you a couple of seconds. The cover teacher can pause this and then play once you're all ready to move on to the next food test. So our next one, we're thinking about our test for sugars. So we use Benedict's test for this. Okay, so the Benedict's reagent. So during this, you want to see the test tube change color. Okay, so it'll go from sort of a normal blue color to green, and then it will go from that to either yellow or brick red. So this all depends on how much sugar is in the food. So if there's a lot of sugar, you can see it go that dark red color, that brick red. If there's a small amount, it will go sort of a blue greenish color. And then if there's a decent amount of it, sort of a middle amount, there will be a yellow color. So once again, you can pause this now, ask the cover teacher if that's needed, write 2 to 3 little bullet points and make sure you include those colors of what the positive test would be. Our third test, okay, we're thinking about proteins. So this one we use Burette solution. So once again, we've got our test tube. You can see the food sample is labeled. You add Burette solution. If you remember from previously, we have to add that and then gently shake it. If there is protein present, you will see there is a color change. So you will see it goes to a dark sort of purple lilac color. So once again, based off this, raise your hand if you need the cover teacher to pause it. Make yourself 2 to 3 points for this type of food test. Make sure you label each one with what test you are going for. Okay, make sure you know which food type and the name of the solution being used. So for this one, for example, would be protein and Burette test. Our last one, we are testing for fats. Okay, so again, we've got our test tube with our food sample. With this case, we're going to use the Sudan 3 stain solution. Similar to our one for proteins, we're going to gently shake that tube. If there are 2 layers that separate and one of them goes red at the top, that means that lipids are present. Okay, so make sure, once again, 3 key points, write down which test we're going for. The cover teacher can pause this now if needed to make sure that all of you get the relevant details down. So our next task is a multiple choice quiz. What I would like you to do is write down just the number, okay, and then the letter of what you think is correct. So number 1 is asking you, which of the following is an indicator used for a food test? Do you think it is iodine, A? Do you think it's B, universal indicator, or do you think it is C, water? Write down the letter which you think. Number 2, what food type turns brown if present after being tested with iodine? That should say, I apologize. Do we think it is A, lipids or fats? Do we think it's B, starch? Or do you think it is C, protein? Number 3, what food test reagent do you use to test for proteins? Do you think it is A, iodine? Do you think it's B, Sudan 3? Or do you think it is C, Burette's? Once again, write down the letter you think it is. Then lastly, number 4, what food test reagent do you use to test for fats? Is it A, Sudan 3? Is it B, iodine? Or is it C, Benedict's? The teacher can pause this now for 30 seconds or so. Write down and make sure you've got an answer to all of these 4 questions. Once you're ready to move on, the teacher can then press play again and the answers will be up on the board in a second. You can see the answers, okay, they are in bold. So number 1 was A, number 2 was B, number 3 was C, and number 4 was A. So make sure you know how many you got of those correct, and then based off that, there are some questions for you to complete. So if you got 1 question right, I want you to do the far left box. If you got 2 questions right, I want you to do the middle box. And then if you got 3 or 4 questions right, I want you to do the last box. If you finish your task before, I'm going to give you roughly 4 minutes to do this. Okay, the cover teacher will time that. I want you to just move on to the next level. If you are on the last one and you already finished, okay, what I would like you to do is write a very, very simple method, okay, on how to compare each of these, or maybe how you would compare results from a number of different food tests. Okay, so think about 4 or 5 different food types, okay, that you'd get maybe in an average meal, and then write down what you would expect to see as positive for each type of food test. The cover teacher will pause this now for roughly 4 minutes before they move on to the next part of the lesson. So this next part, you can see this is a tick box exam question. So an athlete decides to try a new type of protein drink after he exercises. The athlete tests a protein drink to check it actually contains that protein. Which solution is used to test for protein in the drink? What I'd like you to do is just write down which one of these you think it might be. So it's either going to be Benedict's, Burette, iodine, or universal indicator. Okay, give yourself about 10 seconds, write down the name of the one you think it is. And the answer to this one, of course, is A. It is the Burette solution. The next question I'd like you to do is some students tested a red cabbage leaf for starch. So they boiled it, they rinsed it, and then they added the reagent to test. So give one safety precaution that the students would have had to have taken in this test. Okay, so you just need to answer that. A, that's only a 1 marker, so you only need one thing. And then B, which reagent is used to test the boiled leaf for starch? So do you think it's Benedict's, Burette, or iodine? So question part A, make sure you write down one safety precaution they'd need to take, thinking about boiling, what that might entail. And then B, which reagent are they going to use? Give yourself about 30 seconds to answer this. The cover teacher will press play after those 30 seconds so you can mark your work. Okay, here are the answers. So you could say goggles or safety glasses, eye protection. If you say gloves, that does not count. If you talked about maybe using glass rod or tweezers and keeping ethanol away from the flame, then that also would have given you marks. Then B, the answer to that one was iodine solution. Our last question for this lesson. So why is a high protein diet useful to an athlete? So you've got to think about one of these boxes. Normally you would tick it, but what I want you to do is write out that sentence you think it might be. So do you think it means it provides amino acids to make new muscle? Do you think it provides fatty acids to produce urea? Do you think it provides glucose for energy? Or do you think it provides lactic acid for anaerobic respiration? Thinking about the words that are used in these sentences, okay, and then think about what these things are, okay, and what food group they would go to. Remember, glucose is not protein, okay, it's a sugar. What I'd like you to do is write the answer, which you think it will be for this question. Okay, and here is the last part of the lesson. All you have to do is give yourself a green pen tick if you got this correct. If you did not quite get this correct, what I want you to do is make sure you know the answer to this question. So just to round up what we have done today, we have stated the positive results for the 4 tests. So we know what each positive test for our carbohydrates, our sugars, our proteins and fats are. We have described how to carry out each of those tests, and then through those questions after our multiple choice, if you got to that last box, you had to compare some of those methods for all of the different food tests. Well done. I look forward to seeing your work and you next week. Have a good weekend.